Thank you so much, Nicole, for the introduction. Tim, are you there too? Good. Tim and I are going to tag team our presentation. So, and I think part of that is because when you were listening to our introductions, you might have been wondering, like, what are these two people doing together? But it's because we're really interested in trying something out that we think is pretty cool, which is building technological innovation. And while you're doing that, also trying to figure out exactly where that innovation might land, what considerations there might be sort of within society. So we're going to talk to you about that. Someone is calling my office phone, which never happens. So I'm just going to hang up on them. There we go. So I'm Siobhan Braybrook, and Tim Malloy is also here. And we're sort of part of a wider interdisciplinary team that we've put together on this particular project. So previous work in my lab in the science side was done by Marina Lenardich, who was a postdoc who's now moved on, and we're going to be joined soon by Stephen Bouter as a new postdoc on the project. Our collaborator in France, Bénédicte Charrier, she's going to be here on sabbatical in about a month. So that's going to be a great addition to our Nobel Foundation project as well. Here's Tim and Greg. Greg is a law student who's working with Tim, and he also has a biology background. This is our collaborator, Sergey, who's at USC, who, as you will see, works in giant kelp. We have a bunch of exceptional undergraduates who work with us on this project, both in the science and on the policy side, and two incredible young women who are working sort of on the more creative art side to both understand what kelp means within community, but also share what kelp means within community. So, Tim, was this going to be you, or is this going to be
Yes, this is going to be me, but okay. You'll have to control the slides for me.
Got it.
So, you know, we call this project Kelp Magic. And the reason is because kelp itself does have some magical qualities when you think about it. So think about this. Not only does kelp not require irrigation, which is particularly important in our extreme drought situations, it actually cleans water, right? Likewise, kelp doesn't require fertilization. Rather, it becomes fertilizer, a more sustainably produced fertilizer that can be used to support agriculture. And it can also be commercialized ultimately for use in bioproducts, biofuels, and food. And beyond all that, it can offer a substantial help in terms of dealing with climate change in its capacity for carbon sequestration. So the project itself is sort of directed at how do we leverage all these terrific qualities of kelp and do it in a way that is both socially and ecologically sustainable. Back to you, Siobhan.
Thanks, Tim. And just if you're wondering also, maybe again, how we got together, that was through the UCLA Sustainable LA Grand Challenge, where Tim and I were part of a team looking at how we could leverage marine resources to help build a more sustainable future, which is how we started developing this project. So as part of that exercise together, this wider team, we sort of imagined what kind of impact we could have. And I'm grateful that we had an incredible introduction to climate change from Yu Zhang before, because we don't have to necessarily dive into that. We can just say, let's imagine that we need to do something about it. And so we did a little bit of imagining just for Los Angeles, what type of impact sort of improving kelp growth, especially kelp growth in a changing climate, could actually have. And so this is a sort of dream that we came up with, but we'll start over here with carbon sequestration. So for example, if we were able to leverage existing here in orange kelp stands around the LA area, but also maybe engineer some new ones, and we could improve the growth of these organisms, we could get at least 8 million metric tons of carbon sequestered each year, which would be enough to offset all of the LA Department of Water and Power CO2 emissions for a year. So this is one of those problem X, solution Y things that Yu Zhang mentioned, but we could do that. You could also then use the kelp, as Tim was mentioning, so you can use it to improve your economy. So a kelp-based bioeconomy could be worth up to $85 billion over the next 5 years. You can also use kelp for biofuel production. And again, we could actually potentially, with more work, kelp could help us produce biofuels that could offset most of LA County's gas and diesel if we weren't already moving to much better sources of energy for our vehicles. But we could also, for example, if we place kelp stands near the water treatment plants or water effluent plants that we have, for example, here and here in the Long Beach Harbor, we could actually improve the water filtration of effluent by removing excess phosphorus and nitrogen completely from the effluent, again, with a correctly engineered stand. So we have a lot of ways within which kelp could be used to improve the situation locally in Los Angeles, but also globally. So they're an incredible natural resource. But as I've mentioned a few times, they are struggling because they are also subject to the warming of our oceans and the acidification of our oceans as products of climate change. So while they could be an exceptional resource, they're also suffering. So we need to fix it. We'll also note for you that kelp are keystone host species ecologically, and their presence actually defines our ecosystems. So it's not just about utilizing kelp and saving kelp so that we can do things for ourselves, but also so we can do things for our wider ecosystem and world. So as a small background for everybody who may not know what kelp or algae are, oftentimes we think that algae are plants of the sea and seaweeds are plants of the sea, but in fact, they're not. So plants sit sort of over here in the tree of life, land plants, and there are green alga and red alga that sit over here. We're down here, animals, fungi, all these different multicellular organisms. But it's actually over here in this branch of life where the seaweeds and kelp live. Most of the brown algae is another way that we call them. And so these large multicellular brown algae are actually most closely related to diatoms, but they're also related to slime molds, which people might be familiar with. And so sort of says, basically, all the things we know that happen in plants or animals or fungi or other multicellular organisms, we can't be sure that any of those are true over here in brown alga. So they evolved about 150 to 200 million years ago, and their multicellularity, this thing that makes them huge and grow really big and be able to pull all that carbon out of the air, but also serve as these foundational species, is a different innovation than when it happened in plants or animals or fungi. And the earliest fossil evidence actually comes from California, which is this Miocene deposit in Monterey. So they've been around for quite a long time. We still know very little about them. We do know that they have very beautiful growth forms. And so as a developmental biologist, which is one of my backgrounds, I'm pretty interested in how they pattern themselves, but also how they grow. And so understanding how these pattern and grow would allow us to, again, leverage them for ecological and human use. So this is just a blow up of that particular area on the last tree that said brown alga. And so over here where we have laminaries, these are the giant kelps that you might be familiar with seeing off the coast of LA. There's also a lot of different types of brown alga and seaweeds that we can find and use. And so, for example, while we know very little about their development in molecular biology, with only 2 mutant genes known, and those mutant genes are mutants with known genes, are actually in this little class over here called Ectocarpus or Ectocarpales. These tiny little fluffs are not to scale compared to the giant kelp. So I'm going to show you another slide where I attempt to show you scale. But we can do something in this sort of tiny lab rat of a brown algae, of a seaweed. But one of the things that you'll see we'll want to do is translate some of our knowledge from this sort of small lab-based species into these giant kelps for their potential uses. So just for your information, there's only 2 published brown algal genomes in the world. We have 1 draft genome for fucus, which is down here. And our collaborator at USC has 1 for the giant kelp here in Laminari. So it's not as if you're standing, looking into a tree of organisms where we know very much at all. And we're just beginning to get into the molecular biology of these organisms. So for something that has such great potential to be so unexplored is one of the things that we would really like to work towards solving. The seaweeds are really this unknown frontier. And if we want to be able to leverage the brown algae, including the kelps, for sustainable solutions, we also are going to need to understand their gene function relationships. And so that is why we're here today, because we need technology development and innovation. And so oftentimes we think about having an awesome sustainable outcome over here. So this is a beautiful kelp forest, a picture of a beautiful kelp forest. Imagine all those things that it could be doing for us and for the environment. And we think, okay, well, we're going to develop some technology and that's going to lead us right over here and everything is going to be gorgeous and beautiful and easy. But I suspect that anybody who's tried to do this before recognizes that there are often all kinds of unexpected blocks and roadblocks that come up even once you've developed your technology to actually implement them to reach that awesome outcome that you're going for. And so that's why Tim and I are working together to try and include a study of what kind of context, sort of social, legal, political context within which this innovation would land, but also being able to make decisions based on our values and the values of our community. Tim, did you want to add in on this?
Sure. I just want to say this is what is, like, for me, so exciting about this project, and I think also important in terms of, from methodological perspective, the intersection of science and policy and law. It represents this unique opportunity to meld concerns about law and governance with a developing technology from the very start, right? So when Siobhan talks about context and thinking about legal, social, economic, environmental, cultural, all these impacts, I mean, we care about this for a variety of reasons. One, as she mentioned, is there's a history of new technologies as they move from kind of basic science into applied science into diffusion, that they run up against roadblocks because of these contexts. So one focus would be to think about that in advance and try and smooth the deployment of a new technology, especially an environmentally and socially beneficial technology like this. But context isn't just instrumental about how do we get smooth deployment. It's also about how do we incorporate these kinds of impacts from a normative sense. That is, how do we think about moving technology in a direction that doesn't run roughshod over the social and cultural and economics of it all, but rather acts in a complementary fashion. So context is important from that standpoint. But also, as Siobhan mentioned, in terms of this project, we think it's important because it represents an opportunity to show how scientific and commercial development could incorporate notions of social values beyond the value of capitalism. So how do we get this awesome sustainable outcome, but do it in a way that is inclusive, broadly speaking, that takes into account issues of social and economic and cultural equity, and also kind of integrates into all that notions of sustainability. Siobhan, can I get the next slide?
Yeah, can I say one thing before we go on too?
I suppose you should. We're supposed to show how collaborative we are. So,
but I spoke too much earlier. No. I think one thing that's important for me to mention, I can't be sure like who is in the audience, how many trainees might be listening or postdocs, for example. But I had a lot of uncomfortability thinking about commercializing science in a way that was just about making money, making money for me or for other people. That wasn't really something that sits well with my own value system. And so it's been pretty amazing for me to get to meet Tim and start thinking about how we can do technology development, we can do innovation, and there's a way to do that that actually aligns with your own values, whatever those may be, even if they're not exactly the same as the values that you always see represented around you. So I just want to shout that out. Okay, next slide.
Terrific. So this project, in a sense, tries to operationalize those concepts we're trying to link together the technology development with a notion or a process or method that's known as anticipatory governance. So think about anticipation. What we're trying to do, as Siobhan talked about earlier, is imagine kind of the life cycle of this technology from initial development, field studies, out to ultimate deployment as a central part of Southern California global economy. How do we get there? What do we have to do today to ensure that smooth deployment and that context is properly taken into account? So anticipatory governance, which I'll talk about a little bit more later, essentially has three concepts that are embedded in it. One is to include the voices of all those who may be potentially interested or impacted by the development and use of this technology, get them involved early on. It also tries to look forward to identify likely scenarios of use of the technology, and then from those scenarios to bring that back in a method known as backcasting to anticipate what will be necessary in terms of legal, social, economic interventions and policies so as to guide development through the basic science into the application of that science into commercialization.
So to look at how that integration might happen between these sort of our two areas and disciplines, we thought we could break it down into these three major goals for you. So first, sort of on the scientific side, we're talking about rapid genetic engineering optimization in kelps or brown algae. And then on Tim's side, concurrently, we would be looking at what the legal, social, and cultural landscape the innovation would land in. And then together, those things would be combined to help us then translate the methods and technology and be changing as we're learning down below into these more useful species. I'm calling them utility species, so these giant kelps. And our overall goal is to, Tim's going to describe for us.
So if you think about, there's really two things we see happening in the long term when this project moves forward. One is obviously to make sure that we're enabling a key technology here that will be transformative in terms of the various ways in which kelp could be used. But beyond that, also thinking about how this approach to technology development could be transferable to other folks who are engaged in genetic engineering with kelp, with other organisms, and maybe also with other kinds of technologies. And that we'd be able to, by using this as a starting point, provide services to assist those who are developing and deploying technologies to work through an anticipatory governance frame. In keeping with this notion that what we do ought to reflect the values, the goal would be to have a startup, what's known as a beneficial corporation, sometimes also known as a B corporation. And this is a creature of law that is sort of an evolution beyond the typical technology firm that not only has in mind, expanding the use of the technology and generating profit, but also incorporating general public benefit as a core focus of the corporation along with profit. So the idea of a benefit corporation is that it has a material impact on society, and its value goes beyond making profit, and it also has a commitment to engagement and transparency. And if I can just add, if you're like thinking, well, what are these like legal and social and cultural things you're talking about? What is the context? What's that look like on the ground, or I guess I should say in the water? What would that look like? So think about just even the early stages of development. Right now, Siobhan is talking, as she'll mention in a minute, about work in the lab to start to optimize genetic engineering, but eventually that'll make its way out into the real world, right? You're gonna have to test it at some point. And if you take a look at, all the regulations that we have out there to deal with new technologies and new materials, there's virtually no guidance as to how that would work with genetic engineering of brown algae at all. EPA currently regulates the use of genetic engineering with respect to green algae and red algae, but specifically excludes brown algae from that. So how does somebody who's trying to develop this and thinking about some of the environmental and health issues that might arise when you start messing around in nature with the genome of a foundation species, who do you go to figure out how to do that and get approval from? Those are the kinds of questions we're asking about in the early stages of the project.
So we decided just to show you our project timeline, because that's sort of a good jumping off point to show you where we're at. Sorry, and I forgot to mention, if anybody else who's listening or part of CNSI is involved in or has thought about that type of B corporation as well, we would love to sort of touch base and hang out and talk about it. It would be really cool. We thought to bring in some speakers to talk about what that type of benefit corporation would be like, maybe as an event at CNSI. So anybody, if you're interested, please let us know. Or if you have experience or know people who do, we would love to get in touch. So we have the sort of science side and the governance side, and as you'll see, they are knitted together. I'm just going to tell you a little bit about where we are with the science side. So I briefly mentioned earlier, we're going to use 2 algo systems to start with. And there are sort of, I guess, scientific and technological reasons behind that. So ideally, we would love to get our technology working in Macrocystis pyrifera, and that is definitely the goal in the second year of our project. And so Macrocystis is the giant kelp, as I mentioned. We've seen beautiful pictures of kelp forests. That's mostly Macrocystis. They can grow up to 100, 200 feet in length, growing like 2 feet a day, which is pretty darn close, if not faster than bamboo, one of the most sustainable land crops that we can think about. But they're really difficult to grow in vitro. As you can imagine, you might need like an Olympic diving pool in order to grow these, plus wave cycles and salt water and microbiomes. They have an annual reproductive cycle, which can make, you know, some of these technologies and future breeding things a bit more complicated. And there are existing rope culture methods for open cultivation out in the water, but as Tim mentioned, that could be a little bit tricky to be working with at the beginning of this project. And so we're actually working in our little lab rat algae, Ectocarpus siliculosus. And this is also not to scale. I tried, if I wanted to do these to scale, where one is 1/2 to 5 inches tall and one is 150 to 200 feet, I couldn't even see the dot on the slide that would have been Ectocarpus. But it's a very small filamentous little tuft of an alga. It is a sort of model species for the field, even though the field is very small, and that's because it's easy to grow in vitro. We can induce a reproductive cycle anytime we want by changing light conditions, and it is one of the first sequence genomes that exists for brown algae. So we have a lot of really useful tools for us to start optimizing our technology in this system before moving it over to the giant kelps. But Tim's going to go for this a little bit more.
Well, yeah, I got excited on
yourself.
Yeah, I was so excited but anyway, so let me just kind of wrap up on this point, which is, you know, even as we're like starting on the lab rat, you have to be starting to think about what happens next and where you're going to go. And one of the things we're looking at, I'll say about a little bit about this later, is starting first, you know, who regulates, you know, naturally occurring kelp when you're trying to use it in a commercial setting, you know. Then you run into the next question is, okay, who's going to regulate genetically engineered kelp, both the process of doing it, experimenting with it in the natural environment, and then ultimately using it in a commercial process? How do you figure out what the ecological and other impacts are? And then the other thing that comes up, and something you have to start thinking about, or that we are, I think, fortunate to be able to think about this early in the stage, is, you know, whose are the voices that we should be listening to, and what is it that they will say? Even now is the perfect time to start thinking about those issues.
So where are we now? About, I guess, my group started in Cambridge about 8 years ago, and we started looking at brown algal development at that time. And we're dreaming of having ways of manipulating genes to understand growth and development in brown alga. And so about 4 years ago, we started working on trying to get CRISPR-Cas9 genetic engineering to work. And so I'm going to show you some preliminary editing evidence in our lab species, Ectocarpus. And we have some draft methods for this. This was basically the work of sort of one person, plus me a little bit, working over the past 3 or 4 years. And so we're grateful for the infusion of the Nobel Foundation Award to allow us to have more people working on the team. So in order to change genes, and you could imagine all the reasons you'd want to do that using CRISPR-Cas9, you have to get it in there. And so we've been able to successfully get fluorescently targeted Cas9 here, targeted within cherry, into the nuclei of Ectocarpus filaments, which is where these arrows are pointing at in A. We've also been able to phenocopy one of the 2 existing developmental mutant phenotypes in brown alga. And that is this mutant in Ectocarpus called étoile. And it's called étoile because the cells don't elongate very well. And so you end up with this, instead of like a beautiful flowing tuft of alga, you end up with these tiny little star-shaped constellations of algal filaments. And so we've been able to successfully phenocopy the étoile phenotype by targeting the étoile gene in a wild type Ectocarpus. And that was work with our collaborator in France, who will be here next month. But our current methods are pretty slow and laborious. They're all manual by hand. And so we need to develop higher efficiency and higher throughput methods. So we have some ideas of how we could do this using nanofabrication, for example, but we would love other collaborators from the CNSI community. So if you're listening and you feel like you might have a cool way to help us really up the ante on how we can deliver Cas9 into these particular organisms, there's all kinds of really exceptional challenges, but we think it's worth it. So if you want to talk about it, please let us know. In Macrocystis, so this is the giant kelp that we'd really like to go to, our collaborator, Sergey Knudsen, there has a lot of now eQTL-based gene targets that they'd like to look at that they think regulate giant kelp growth. And they also have a spore library of Macrocystis from throughout the coast of California, and I think even up into Washington now. So we have the material we might want to edit in, and we have ideas of which genes we would like to go to. And then Tim is going to tell us a little bit about what progress we've begun on the governance side of things.
Yeah, thanks, Siobhan. So just quickly, in terms of governance, you can see from the timeline here, we've really got 4 foci. So one is, we talked about who are the voices and what are they saying. So we have a stakeholder survey that's going to look at a variety of populations in the LA region, focusing on, their knowledge and expectations with respect to genetic engineering and also use of kelp. The thing we're working on that will, kind of bridge all the other stuff is thinking about the policy and legal issues associated with the use of genetically engineered kelp, both from, basic science, applied science, commercialization. And then lastly, we're focusing on these other pieces of anticipatory governance, developing the methods and templates that would be used to both create the scenarios and then to backcast those so that we can start thinking about how to develop, both influence the course of the scientific work, but also start thinking about the course of policy and outreach that's needed to reach the vision that we have for sustainable use of kelp. The other thing I just want to say about this is that a big part of anticipatory governance is integrating at a granular level the science and policy, to the extent that we've already started now having the legal team participate in the lab meetings for the science team. So that's a very important part of it. And we've also already started the survey of what the current legal, economic, and cultural contexts are for the use of kelp. today. So, I mean, the big question that you may be wondering is, how are we working together? And no, you're perfectly fine advancing slides there. How are we working together? And I'll just say there's 3 things that are going on. I already mentioned the research team integration. We're also looking for many voices through multiple vehicles. So the stakeholder survey is just one. We're also doing outreach to indigenous communities. And we're also creating a seminar series to bring speakers to UCLA from a variety of disciplines, all focused on this issue of anticipatory governance and the science of genetic engineering. And we're also working on developing approaches for decision-making in the development that are very explicitly and intentionally value-based decision-making processes. And I guess the only question is whether there are questions.